moving, switch to the slide again. Yes. Great. To our third speaker of the night, Dr. Jonathan Woodsmith. So Jonathan has been working at Indivumed and actually with Biognosys for almost 4 or 5 years, right? So Jonathan graduated from with a Bachelor of Science in University of Leeds and a Master of Science and PhD from University of Liverpool. He then moved to Germany to join the Max Planck Institute for molecular genetics, and then had also a short stay in the University of Graz in Austria before moving to Hamburg to join Indivumed, which now just became Indivumed Therapeutics. So Jonathan will gonna talk to us about how they leverage omics data and actually getting actionable insights and value out of this data. And without further ado, I will hand it over to Jonathan.
I already have one. Thank you. So thank you very much for the invite, Biognosys. As was said in the introduction, I've been working together with Biognosys for almost 5 years now, and can only recommend. But Indivumed, who are Indivumed? Indivumed is actually it's not a particularly new company. It's actually 22 years old now. Was founded in Hamburg, and for the first 21 years of its existence was actually focusing on high quality biospecimens, and was selling either the tissue surrounding that or services based upon it, so IHC, qPCR, and so on. And one of the key things that Indivumed was doing was trying to keep the quality of the tissue as high as possible. And what they realized at some point was that they have a biobank of ridiculously high quality tissue which they could generate multi-omic data for, and then develop a discovery database for which we can find therapeutic targets, for which we can find biomarkers, etc. And it's actually a side project of this that I'd like to discuss a little bit today, which is to do with immunopeptidomics. But first, taking a step back, what is the USP of Indivumed? It's the way that they collect the tissues. And so we've heard 2 talks today which are focused around using FFPE slides, which is obviously a clinical standard. With FFP slides, you obviously have the tissue kind of lying around in formalin for quite a while. And one of the things that Indivumed was founding on was actually trying to standardize tissue collection so that that wouldn't happen. So that essentially, as soon as the resection is complete, you start the clock and you have cold ischemia time. So it's where the tissue is lying on ice. And that cold ischemia time is one to be reduced to 10 minutes before you freeze. On average, in the database, it is around 10 minutes. We actually have shorter than that. We actually have longer than that. Very much dependent on the type of surgery that you've undergone. Some surgeries are much more complex. But this does mean that we actually have nurses or assistants within the surgery that are trained to then take the tissue and freeze it in liquid nitrogen, so this is fresh frozen tissue, with a goal of 10 minutes. And what's the reason for that? So, I mean, if you're looking at multi-omic data, so the genomics will stay stable. You can have that in fixed formalin, that's fine. The further you get away from genomics, the more dynamic the signal will be. And so here's just an example. This is an IHC assay, and this is tissue that has been on ice. So this is cold ischemia time, and you can see that the signal increases from 20 to 60 minutes. This is actually phospho-ERK, so this goes up with ischemia time. I was surprised as well when I found that out. And what this does show is that the longer that you have the tissue lying around, the further the complex molecular data is from the disease as it was in the patient. And this essentially is the entire USP from Indivumed, is to be able to collect tissue in a standardized manner that represents the disease as closely as possible. And this is done through a clinical network that is worldwide, and so mainly focused in Germany, but we do also have clinics that we work actively with in Italy and in Spain, as well as Asia and Brazil, and we're going to re-expand the clinical network also to North America in the coming years, hopefully. And so the idea is that we take the patient and then we get the resected tissue. So we have both normal adjacent tumor tissue as well as tumor tissue for the majority of cases. I'll come to a couple of exceptions. And then we have multi-omic processing. And so we go for whole genome sequencing, microRNAs, transcriptomics, proteomics, phosphoproteomics. We have some pilot projects involving lipidomics as well. And we also have H&E images from the tumor tissue as well. And we do the data processing and collect all of this database, collect all of this multi-omic data as well as clinical data. And this is really key because the multi-omic data is actually very valuable in and of itself, but what makes it especially valuable is if you have the clinical data and the clinical follow-up data. This allows you to then say which molecules are then going to be associated with prognostic signals, as well as associating with certain histological subtypes, etc. All of the things that you can then collect from clinical data. And we do this also, the clinical data is also collected longitudinally. So we have an aim of collecting clinical data for 5 years. Not always reached, obviously, but we have some data in the database that goes up to 10 years follow-up data. So what are the tissues that we have in this database? We actually have a focus on colorectal cancer and lung, as well as having reached, I think we're over 500 cases now in breast. A particularly interesting data set that we have is actually about 300 metastatic samples from colorectal cancer. These are predominantly metastases that have landed in the liver. And we're also expanding out the cohorts that we have into pancreas, ovary, bladder, prostate, etc. And for the vast majority of these, we have adjacent normal tissue, with the exception being breast is actually very difficult to get adjacent normal tissue from. And it's certainly very difficult to get proteomic and phosphoproteomic data from breast tissue, and metastatic tissue as well. Obviously, if you're resecting something from the liver, getting adjacent normal tissue from the colon is surgically unlikely. And so what do we do with all of this data? It is essentially a discovery machine. We then have a multi-omic data set with which we can go and find biomarker discovery, as well as therapeutic target discovery. We actually also run services for in silico biomarker or target validation. So if companies have a specific molecule of interest and they want to de-risk a project by seeing what that looks like in an independent data set, they come to us and we say, okay, this is, yes, it is more highly expressed in late stage cancer. It's actually associated with histological type X, and it is or is not associated with prognosis. These are all questions that we can answer in an independent data set. These have often been observations that have come out of years of academic work or looking systematically at TCGA. It's always very good to have an independent validation. So this is one of the services that we run. But the main focus of the company going forward is going to be on discovery and validation. So this would be therapeutic targets as well as biomarkers discovery. And so the majority of the work that we've been doing is looking to identify and validate so what you would kind of classify as classic therapeutic targets, membrane receptors, protein degradation targets, not just PROTAC targets, but other modalities in the protein degradation field, I think is very interesting, and also looking into enzymatic tumor drivers. But one side project that I'd like to discuss today is actually using proteomics in a completely different manner to the whole proteome profiling or the phosphoproteome profiling that we usually do, to try and address therapeutic targets which are outside of these kind of classic therapeutic target definitions. And that's looking into the world of neoantigens or tumor-associated antigens. And so as a brief intro into this, this then starts to come into the world of the immune system. And so we all know that cancer is not just a disease of the tumor itself, it is a complex disease of the tumor microenvironment, consisting of stromal cells, and immune cells, etc. And this plays a massive role, hence the huge growth in immune oncology treatments that has been exploding in the last decade. And very briefly, what happens is you have the tumor. I'm just going to see if I can switch this to pointer. So as tumors die, they release antigens into the surrounding tissue. These antigens are recognized by the immune system, often things dendritic cells, or antigen-presenting cells, which activate then T cells. T cells then migrate back to the tumor, and because they have been activated by these specific antigens, they can then recognize the antigens on the tumor, and the immune cell then kills the tumor. All very nice in principle, doesn't always work, hence immuno-oncology treatments. But one of the burgeoning fields in immuno-oncology is essentially being able to try and weaponize this cycle. So we know that the cancer can try and turn this cycle off or tune it down. And so one of the things that we want to try and do is can we actually take advantage of what biology has already evolved to try and kill the tumors. So really weaponizing this immune response. And there are actually many, many different ways in which you can do this. And they all have the neoantigen or the tumor-associated antigen kind of at their heart. So we can do things like we can either engineer CAR receptors or T cell receptors to actually be able to better recognize the tumor cells for more efficient killing, or have bispecific antibodies which actually facilitate the interaction between the immune system and the tumor cell to be able to then make more efficient killing, as well as something I think is particularly interesting is generating vaccines for tumors, which would be then patient-specific DNA, RNA, or peptide-based vaccines, which will activate the immune system above and beyond the basal activation that any given patient already has. And what would this look like, if you excuse the slightly poor diagram, essentially from an individual patient. And so often when we talk about precision medicine, we're talking about groups of patients because it's not economically feasible to generate drugs for an individual patient. However, when we think about vaccines, which in principle you could just generate by printing RNA, this is certainly something where you could take precision medicine to an individualized level that targeted therapies are very unlikely to reach. And so what you would do is you would essentially take the tumor and you can then sequence it to find neoantigens. You could then predict which ones are actually going to be able to activate the immune system and take those into a very quick experimental setup in the lab, something like an ELISpot, to be able to see which ones activate, so which neoantigens can activate the T cells. You can then essentially print these and then put them, take the immune cells, activate them, put them back into the patient, maybe even combine these with checkpoint blockades, and you have a treatment cycle which is actually feasible within the lifespan of the patient. You have slightly more involved techniques which involve taking the immune system out of the patient, engineering it, and then putting it back in. One of the main things that's the problem with this at the moment is the money and the time that it takes to actually do this engineering. This technology will no doubt get better within the coming years. But this is all to say that there are many different ways of being able to do this, but the point is that you can really maybe even get down to an individual patient level with cancer treatments. And so if we look at tumor-specific antigens, where do these things come from? So classically, you would think of these things as maybe either SNVs or indels. And so these are then when there is a protein coding mutation that is introduced within the cancer. And these protein coding mutations would then obviously result in different peptide sequences, which are then presented by the MHC at the cell surface, which could be then recognized by the immune system. But there are actually many, many other types of tumor-specific antigens or tumor-associated antigens. And without going through all of these, the ones that I'm going to focus on today is actually coding long non-coding RNAs, which sounds like an oxymoron, but it isn't. It's actually quite an interesting field. And one of the reasons why this is interesting, actually, and this is just a paper that was published a year ago from Oxford University, is the peptides that are produced from long non-coding RNAs have been shown to be immunogenic and drive a potent anti-tumor response. So what this means is that these coding long non-coding RNAs have been shown in mice at the moment, not in humans, to be produced in cancer contexts, stimulate the immune response to actually slow down the growth of the tumor. And so this is something that we thought that was very interesting. And so can we then use our multi-omic data set to be able to define and identify coding regions of long non-coding RNAs that we can then see in patients? just as one step back, what is a long non-coding RNA? Long non-coding RNAs, there are thousands of these that are annotated in the human genome. And majoritively speaking, they have been shown to be regulating transcriptional patterns or protein activity. Very commonly, so more commonly in the recruitment of epigenetic regulators. Exist would be the most common example of a long non-coding RNA, which actually turns off a lot of the gene expression on the X chromosome. But there are actually multiple functions here. So you can also act as a sponge for microRNAs to be able to then turn off microRNA functions. And these have been shown to have multiple cancer-related functions. And when we look into our data set, if we look across the 1,000 primary CRC tissues that we have, we actually see thousands of long non-coding RNAs that are deregulated in cancer. And we also see this as well in the gastric cohort that we have. And one of the advantages of the data set that we have is that rather technically, we haven't used poly-A enrichment for our RNA sequencing. We used rRNA depletion. The advantage here is that 50% of the long non-coding RNAs are not poly-A tagged. So if you use poly-A enrichment, you won't identify them. And so we actually get increased sensitivity by using rRNA depletion here to be able to really sensitively identify things that are deregulated in cancer. We can actually do this across all of the cohorts that we have, and we can do this within subcohorts as well. So you can see here that what is interesting is a lot of the upregulated long non-coding RNAs are tumor of origin specific. So you can see here that there are well over 2,000 long non-coding RNAs that are upregulated in NSCLC, and only shared under half of those across the other 6 diseases. And you can go into sub-cohorts as well. So adenocarcinoma in lung, squamous cell carcinoma in lung is very tissue specific, the origin of these molecular alterations. We also identify long non-coding RNAs that we would expect as well. Just as an example from liver tissue here, we have the link 01419, which is actually not only upregulated when you see it in primary tissue compared to normal, but you also see it much more commonly. And this is actually quite interesting with long non-coding RNAs. In comparison to protein coding genes, they are sometimes actually completely switched off and then switched on again, rather than going up or down. This is something that we want to look at systematically, because this may explain tumor specificity of the cryptic peptides that we later see. So if we go into cryptic peptides, I'd actually never heard of these before I started on this project, but they're a fascinating group of mini proteins. Here is some examples of what they can do, but essentially they're cryptic because they are essentially their source is not from the protein coding genome, although they are proteins themselves. They're mini proteins, usually between 15 and 100 amino acids in length, and they are often sourced from long non-coding RNAs. And just as a very small example here, there's SMIM30 is a microprotein that's actually produced from the link RNA 00998. Long non-coding RNAs have horrible names. They're all just numbers. But anyway, and what you can see here is that when you actually produce this protein from an exogenous vector, you can see that you do see an increase in vitro in the number of cells. And when you then knock down the long non-coding RNA, you can also see that you see a decrease in number of the cells. So this is just one of many examples to show that these mini proteins actually have functional roles in cancer as well, both promoting as well as inhibiting cancer progression. And although they're very small, and it sounds kind of like an oxymoron to say that you get coding regions from long non-coding RNA, they actually appear to be important and are not as rare as at least I was aware of before. And so, but going back to this, what do we then want to do? We want to then define tumor-associated antigens or neoantigens that are the center of this weaponizing the immune response paradigm. And so how do we do that? What we need to do is we need to identify any of these cryptic peptides that are then presented at the cell surface. And this is where then we could work together with Biognosys and their True Discovery platform to be able to enrich MHC1 molecules from the tissue using immunoprecipitation. Originally, we actually also did this using MHC2, which works very nicely as well, but today I'm just going to focus on the MHC1 results that we got. And you can then enrich for the MHC complexes, and you can then elute these from the beads to be able to get at the peptides, and then you throw the peptides in the LCMS MS, and you can then identify, and then you can see which peptides you have identified. One of the huge advantages of DIA is that you can actually custom peptide search very nicely. So if we go in, we can look for proteins that are from the protein coding genome, but we can also then go and predict which proteins or small peptides may we think are present from this non-coding RNA space. And to be able to do that, we use an online database, small-orf function, and they have over 600,000 predicted small open reading frames within the coding, within the human genome. The problem is if you throw 600,000 small ORFs into a DIA experiment, you will find stuff. You really need to, the key here is to reduce your search space as much as possible to the most likely things that represent potential cryptic peptides. And the good thing is that we have the data to do so. So we can go into our CRC data set and we look to see which are the long non-coding RNAs that are then expressed. And this allows us to then combine this with the small ORF predictions to look for 14,500 putative cryptic peptides. And so you can reduce this search space by over 97% through integrating the transcriptomic data that we have with online in silico data sets. This is just to show what this data looks like. So as I said before, the majority of these small ORFs under 300 nucleotides in length, so under 100 amino acids. And the median side is actually around 18 to 19 amino acids long. And when we look at this, what you can see is this is actually a multi-exon long non-coding RNA. And you can see that the SMORPH gene here is actually present in the first exon here of this long non-coding RNA. So they're really, really tiny things. That doesn't mean that they don't have any physiological relevance. And when we look into the peptide characteristics that we get, so when we've extracted these peptides from the MHC complex, we would expect to see a large enrichment in 9 amino acid length peptides, as this is what you would get from the MHC class 1. This is clearly what we see. And when we look to see of those peptides, so this is not only from the, this is including the protein coding genome as well as the cryptic peptides from long non-coding RNAs. What we can see is that the vast majority of the peptides that we identify also have expression support from our multi-omic data set. So this is showing that these things are floating around in the cell and then end up on the cell surface. We can also do a sanity check based on the HLA alleles that we have. So because we have the genomic data from the patient, we can then HLA type them. And each of these HLA alleles has a motif which would suggest that these peptides that we've identified should have certain features. This is only a sanity check. It doesn't take into account HLA binding specificity and the amount of each HLA you have. And so these are only trends that we should be observing. But here what you can see is that at the position 2, we should certainly be seeing glutamic acids. We should be seeing prolines. We should be seeing arginines. And this is what we see in the peptides that we identified. So you can see here glutamic acid is certainly very high, as well as proline and arginines, as well as leucines and alanines. The only one that we don't identify that, as you would predict from the motifs, would be then methionine. And then if we look at the second anchor position within the HLA binding motifs, we can see that we really shouldn't be seeing any of the prolines and the glutamic acids, but instead lysines, tyrosines, leucines, and phenylalanines. And this is indeed what we see. And so you can see a nice separation, which shows that the quality of the data that we're getting from Biognosys' platform is actually very high, and we're identifying peptides that the biophysical properties of the peptides that we're identifying match what the patient's biology would tell us. And so do we identify cryptic peptides? We do. We identify almost 100 cryptic peptides, of which 33 are exclusive to tumor. We don't identify them at all in the normal patients. And so these is then what you would classify as then tumor-associated antigens that are at the heart of the therapeutic approaches. we also see that some of the peptides we identify in multiple tumors, multiple patients. This then becomes advantageous, as you might not need to generate a new vaccine per patient, but you could identify patient groups. This is only 20 patients, so we can't really extrapolate this to larger patient groups. But what we can see is that from the data that we have at the moment, we can certainly see that we appear to see more tumor-exclusive events than normal exclusive events. And we certainly see that some of the peptides are present in multiple tissues. And so within the last 5 minutes now, I'd like to go through then one example of what we've identified here. And this is from the long non-coding RNA SNHG6. This actually has 10 small ORFs that would have a methionine as a start codon. That's not necessarily, doesn't have to necessarily be the case that they need a start codon of methionine. But this long non-coding RNA is actually relatively interesting. It's actually been seen as upregulated in colorectal cancer before. And that's what we see in our data set. So this is from our transcriptomics data. We see that it is much more highly expressed in primary tumor tissue than in normal tissue here. And interestingly, we also see that it's associated with poor prognosis. And so the red line here is when SNHG6 is more highly expressed in the tumor compared to the blue line when it's lowly expressed. And what you can see from this Kaplan-Meier curve here nicely is that this long non-coding RNA is actually associated with poor prognosis here. One of these small ORFs has actually already been functionally characterized. And so when we look at the 5' end of this long non-coding RNA, there is approximately 40 amino acid length protein that has been shown to be involved with increased cell migration in endometrial cells. And so when you put this into an exogenous vector in endometrial cells, you can actually see here that after 24 hours, you see an increase in cell migration in this scratcher assay. The SMORPH that we identify is actually further towards the 3' end. And so this is then when we've taken the immunopeptidomic results from Biognosys, and we've enriched for the MHC class 1. We've stripped the peptides off, and we identified a peptide here that is identified in 2 patients that have overlapping HLA alleles. And we can also see that these actually have very nice mass spec results. And so we can see that you see very nice overlapping here of the MS spectra, and we see good coverage of the peptide sequence from both B and Y ions. Which actually would help us to have increased confidence that this is not spurious peptide identification. We really need to go down and dig into the detail of the data to show that it's actually present on the surface of patient tissue. This is observed in 2 patients with right colon cancer from our data set, so 10% of the patients that we've tested. And you can go into databases online and you can see that actually it's not, this peptide has been observed in lymphoma once before, but in any other normal tissue has never been observed. And so this is almost then your ideal case, where you have something that has been shown that it is on the surface of patients' tumors, but has not been identified in any other normal tissue to date, which is then the definition of a tumor-specific antigen. Which is what I've just said. And so this is just then a very interesting case in point, in that this long non-coding RNA, which already has roles in cancer to do with being a microRNA sponge, may also have multiple roles in the protein coding realm, where several small peptides produced from this long non-coding RNA appear to be produced in the cell, one of which is shown to be functionally relevant in cell proliferation, and the one that we found appears to be then identified at the cell surface. And so, as I said, this is a pilot experiment that we've been running with Biognosys, and we're really at a crucial stage. We've shown that we can use the transcriptomics data to reduce the search space to allow a sensible DIA experiment to be run. We can not only do that, but we can then identify cryptic peptides expressed from long non-coding RNAs at the cell surface of patient tissue. We need to go in and now see if we can validate these using HLA allele binding in vitro validation, as well as crucially testing the immunogenicity of these. So the vast majority of peptides expressed at a patient's cell surface do not elicit immune response because those things are cleaned out, obviously, when your immune system develops. But our hypothesis is because these appear to be more tumor specific, that there may be an underlying tumor response there, which we could maybe boost through using some form of either multiple peptide or RNA, where we're actually able to put the peptide, we're actually able to get the peptides onto the patient's cell surface to boost the immune response and then hopefully slow down cancer progression. And finally, this is also not the only way in which you can look into this data. Because we have the genomics and the transcriptomics data, as well as the phosphoproteomics data, we can then go fishing in this immunopeptidomics data set to see whether we can actually further expand the immunopeptidome. And the idea being that the more comprehensive and the more tumor-specific neoantigens or tumor-specific antigens that we can identify, then the better that we will be able to then use that as a launch pad for developing more either individualized or precision oncology approaches. And with that, I'd like to thank not only the team that have been behind this bioinformatic analysis at Indivumed, but also the team at Biognosys, who we worked closely together with to develop this protocol. I know Marco had a very large role in that. And thank you for your attention.
Thanks a lot, Jonathan, for the interesting talk. Any questions from the audience?
I have just a small question related to the TCR you were showing just before. I've seen that you're using ELISpot. Are you considering running TCR sequencing at large to have the full specificity of the TCR you're targeting with the TAAs or TSAs?
We've already tried it on the data that we have because we've got whole genome sequencing. So you can extract it, but the depth at the TCR locus is limiting. So we're 60x average on the tumor genome, which obviously for TCR sequencing is limited. The idea at the moment is to take this to a minimal viable product, which is to show that we can in vitro bind a HLA peptide to the HLA allele and show that it's at the patient's surface. The ELISpot is something that we're going to do in the future and outsource, possibly. And when we do that, if we can get to the minimal viable product stage, we will probably go down the route of doing deeper TCR sequencing on the probes that we have because we've got more tissue, so we can then send that off. So it's one of many experiments that we can and should do in the future. The goal at the moment is to get to the minimal viral product stage of HLA allele, showing that the peptides bind the HLA alleles in vitro.
Great. Any additional questions? Perfect. If not, let's thank Jonathan once again.